My name is Dario Farina. I'm a professor here at the Department of Bioengineering. I work on neurorehabilitation technology, and today I have the pleasure to host the Bioengineering Department Research Showcase that has been prepared well, in the occasion of the visit of our advisory board, but also for the entire department that has grown over the years and that has grown in activities, in people, so that everybody can listen at the variety of activities that are going on. So we have a very packed schedule, so I will not take more time. I think we should start immediately. For the speakers, you have 8 minutes and 2 minutes for questions. First speaker is Shirin. Just come in. Yeah, and please.
Good afternoon, everyone. My name is Shirin, and I'm a first-year PhD student in Dr. Rodrigo Ledesma Amaro's lab. I'm very happy to share a snapshot of my PhD project with you today, which is focused on engineering anthocyanin production in urolipolitica utilizing CRISPR AI. So, first of all, what are anthocyanins? Why are they interesting? Anthocyanins are a water-soluble class of flavonoids. These are specialized plant metabolites that we find in most flowering plants. The most abundant, cyanidin-3-glucoside, is responsible for the red color we see in most berries, in apple peels, and it's a very interesting compound for the food industry particularly. Due to its multiple bioactive properties, it can function as both a colorant and a preservative. The food industry is able to use compounds such as this to deal with larger systemic issues. For example, meeting the right nutritional profile for products, meeting consumer demands, but also cyanidin-3-glucoside, for example, can be used as a biosensor in food packaging, helping cut food waste, and has also been found to increase the palatability of alternative protein products, which helps deal with sort of the environmental impact of our food production. However, despite all of these promising applications, there have been challenges in its widespread adoption, and this is mostly associated with its methods of production, which currently rely on extraction from plant material. This is a process that is not only unsustainable, but also very costly, and due to the stability of this compound, it also results from very low yields. So one solution is to develop microbial cell factories to produce this compound instead. The idea behind this is that you have a microbe, such as a yeast, you feed it with the right nutrients, such as glucose, and it in turn produces the compound that you want, and you can harvest this. Now, the most important choice when developing a microbial cell factory is your host organism. And for this project, we look at the oleaginous yeast urolipolitica. This is because of its promising industrial traits. It's very robust to a wide pH and temperature range. It can achieve high cell densities. It can use a multitude of carbon sources, including low-cost sustainable options like waste from agricultural production. And it has a safe status from the FDA, which means that we can use it for food applications. In particular, for anthocyanin production, it also has promising traits like a high malonyl-CoA flux, which is a key precursor. It has the right subcellular structures to express plant enzymes. It has a hydrophobic environment that helps with the stereoselectivity of these reactions. A high tolerance to aromatic acids, meaning it's less likely to suffer from toxicity issues with intermediates. And it's been proven to produce one of the key steps in the general flavonoid pathway, which is flavanone. Now, for a glimpse of what cyanidin-3-glucoside pathway looks like, as you can see, it can be broken down into 5 modules. So that means in Eurovia, from its native metabolism, we need to express 13 more genes that come from the plant system in order to produce this product. And from past bioproduction studies, we only see construction until module 3 in Eurovia. In E. coli and Cerevisiae, on the other hand, it's been attempted to produce cyanidin from right upstream the pathway to the bottom. However, the yields have been very, very low. In E. coli, these attempts have not been successful at all. And with Cerevisiae, they still rely on the supply of a very costly precursor. And one bottleneck that these studies have in common is the promiscuity of one of the key enzymes, which results in a lot of flavanol byproduct accumulation. So to achieve cyanidin production in Yarovia, we broke down this project into 3 key objectives. The first is to establish a platform strain for naringenin production, which is the products from Module 3. This can then serve to test different metabolic engineering strategies to achieve integration of Modules 4 and 5. The second objective is to tackle the issue of enzyme promiscuity, and the third is to regulate metabolic flux between growth and production in order to maximize our yields. So as I mentioned briefly before, what we see in the plant system is something like this, which looks a lot like a metabolic grid. We have the same enzymes that can use precursors and multiple steps of the pathway, which leads to the generation of a lot of compounds, but not particularly high amounts of the one that we're most interested in. Now, in the plant system, we don't see the same issue of flavonol byproduct accumulation as we see when this pathway gets introduced into microbes. And one of the hypotheses why is because of the spatial temporal regulation we see in the plant system. So, as you can see here, we have something called a metabolon, which is a loosely ordered complex of enzymes. So one thing we want to test is actually introducing a synthetic metabolon in Yarovia to see if we can actually achieve higher yields of cyanidin by increasing the localization of enzymes and improving the local substrate and enzyme concentrations. This can also help limit the accessibility of certain precursor to promiscuous enzymes. The other challenge we have is to regulate the flux of carbon between growth and production. So generally when we express a plant pathway in a microorganism, we're competing for the same pool of carbon with just growth, which is what the organism wants to do naturally. It's very hard to deal with this issue because all of the genes involved with growth are essential. So if we just knock them out, our cells don't grow at all and we can't produce anything. So one approach that metabolic engineers have adopted over the years is two-stage fermentation, where we have a first phase where we just focus on growth, and then a second stage where we flip to just production. And this can be achieved by many different tools, but one of them is CRISPR AI, which relies on the use of orthogonal deactivated Cas proteins that we can target to our genes of interest, either upregulating their expression or downregulating it, depending on whatever transcriptional domain we've fused to this Cas protein. Now, CRISPR AI has been developed already for baker's yeast, but in Yarovia there's been very few attempts and never having both activation and inhibition in tandem. So to achieve this, we need to work on developing different parts, including an effective inducible promoter that allows us to switch on demand, and also be able to design proper cassettes to target the genes that we're most interested in to decouple growth and production. Now, as I mentioned previously, the first objective was to optimize for naringenin production, and so this is what the past year has been dedicated towards. We've looked at different wild-type genomic backgrounds of Yarovia, looking at overexpression of other native enzymes, and also comparing the difference between random and site-specific integration, which is actually very hard to achieve in Yarovia, and we've developed tools to do this. And what we can see from preliminary results is that we are already producing in the range of 200 to 300 milligrams per liter without a significant burden on growth, which is almost 5 times as much as what has been produced in cerevisiae attempts looking at cyanidin production, which places us in a good position to get higher titers for cyanidin. Over the past year, other things we've been able to focus on through this project is some science policy work. So I've produced a background study for the Food and Agriculture Organization looking at fermentation-associated microorganisms. I've co-authored a briefing paper for the Institute of Medical Sciences and Engineering, and we'll go on to do a postdoc fellowship in the new year. Thank you.
Start with a general one. So you have shown us a plan for the full PhD. Which do you think would be the main challenge, the challenge you're most afraid of?
So I think one of the bigger challenges is actually how to introduce all the different things we want to try in Eurovia. It's an organism that doesn't do homologous recombination, which makes engineering it really difficult. But that's something that we've tackled a lot this year. And so now we have the methods to actually test out the more cool ideas that we have. So.
Thank you. Other questions? There's one there.
Not technical. Is your background study for the FAO published?
It's going to be published in March.
Okay, I'll keep an eye out for it. Thank you very much.
All right. We thank Shirin again. Thank you very much.
Thank you.
And now we change the topic. Nicola, the group of Adam Selitz, will talk about wound healing. Please.
Hello everyone, I'm Nicola. I'm a research associate at the Selitz lab where we are developing a handle by printing device to print click hydrogel for wound healing. So wound healing, chronic wounds do not spontaneously heal, and wound healing is a physiological process that is a response to tissue injury. It's a dynamic process, and whenever one of the dynamic phases fails, the wounds won't be able to regenerate on their own. Failing in regenerating brings wounds to be chronic, and they heavily impact the quality of life of people affected by wounds. This can be linked to diabetes, burns, traumas, and the percentage of people affected by this chronic wound is relatively high, and the economic impact is also high. At the moment, the current strategies that we have to treat chronic wounds are mainly prevent infection, dressings, and scaffolds that are just passive structures that are fabricated ex vivo. So what we are doing is to develop a new technology to promote the regeneration of wounds. We're basically developing a handled in situ bioprinting device, which looks like a pen, I'm going to show you in a bit, that is able to extrude a material, a hydrogel, that can fill the wound, degrade while releasing therapeutics to regenerate the wound. So as you can see from the schematic, we have a wound, we have our pen where two materials are mixed together and extruded. We develop the scaffold, we fabricate the scaffold intraoperative, and then the scaffold degrades and regenerates the wound. Now, the materials that we're using as bioinks, we published them already in the paper you saw in the top right, are bioorthogonally cross-linked click hydrogels. They're made of gelatin, which was functionalized with tetrazine, and alginate, which was functionalized with norbornene. You can see in the NMR spectra here that we successfully functionalized the two polymers. So what happens is that if you create two solutions of gelatin and alginate modified as such, they spontaneously cross-link, they spontaneously react and cross-link to create close-link hydrogels, which are the materials that we are printing. We mixed these two materials at a 1-to-1 ratio, volumetric ratio, and we tested different concentration, 2, 3, 4, 5, and 6%, to investigate what was the most promising as a bioprinting ink. We then tested the rheological properties of these solutions. You can see on the top the alginate and on the bottom of gelatin at different concentration to see if the rheological properties were actually suitable for the printing process. As you can see from the different scale bar, alginate had higher rheological properties, which were useful for us to give stability after the printing process. So this is how our device looks like. This is a schematic representation, while this one is the actual device with the channel. We have the two hydrogel precursors, the gelatin and the alginate, which are mixed. They are injected via two external pumps. They are mixed inside a microfluidic passive mixer, and then they are ejected by the tip of the pen. So you have alginate and gelatin which are injected, they mixed inside the grooves, and then they are ejected as a cross-linked hydrogel. Now, the first thing we did was to optimize the printing parameter using a robotic automatic printer. We tested different parameters, such as the speed which we were using to write the materials, but also the cross-linking time at which we extruded the material. So before being completely cross-linked, during the cross-linking, and then after being cross-linked. We optimized this parameter. We created a library of parameters. We were pre-gel and post-gel at different writing speed, and we were able to optimize the concentration of materials that we wanted and the time that we wanted the material to be printed for. We then moved on and moved from the robotic printing to the manual printing. This is how our device look like. It's a plastic case which was fabricated via stereolithography. You can see here the two tubes that inject the material and they are connected externally to the syringe pump. We have the microfluidic mixer here, and then we have a tube that folds up to then be at the tip of the pen. This is how the pen looks like while we're handling it and working with the material. We recruited 20 volunteers and we asked them to test our pen. We asked them to print the same shape slow and fast because we wanted to check if we had control over the dimension of the filaments that we printed. So by printing at different speed, we would be able to print different dimensions. We then tested the possibility of embedding a therapeutic, so the printed scaffolds would be the structure for the tissue to colonize, but then we want some therapeutics to be in to actually promote the tissue regeneration. So we embedded a model growth factor VEGF-A inside this precursor, and we printed them to obtain a VEGF-A laden scaffold. You can see that in time, our hydrogels are capable of releasing steadily the VEGF that we incorporated. And then we tested the supernatant that were released from the hydrogels on UVEC cells as a model of endothelial cells. You can see as the metabolic activity in presence of VEGF, fresh or released from the scaffold, increased the metabolic activity. Moreover, if you use the supernatant to culture UVEC spheroids, you can see that spheroids start sprouting when we have the presence of VEGF fresh or released from the scaffold compared to the controls. So finally what we did was to add an extra chemistry to our material, which is a muscle-inspired chemistry. So we want to print the material in the wound effect, but we want the material to stay where we print it. So we added an adhesive chemistry so that the scaffold can stick to the tissue. You can see from the video here, we set up a pool of test and an adhesive test to skin. So in the case of the non-adhesive scaffold, the scaffold just detaches from the skin, while in the case of the adhesive scaffold, when we pull out, you can see the scaffold is stretching and then will undergo cohesive failure. We then quantified the adhesive strength, and it more than doubled when we added the muscle-inspired cross-link chemistries. So as a conclusion, we develop a handheld device that can print orthogonally cross-linked hydrogels that can spontaneously cross-linked. The two hydrogel can cross-link inside the handheld device. We optimize the formulation. We optimize the printing parameter both in a robotic and manual way. We proved the possible release of VEGF, and we also proved the possibility of adhering the hydrogel to tissues. And we are now testing further the potential for actual wound healing. So with this, I would like to thank my PI, Adam Silitz, and our research group, and all our funders and collaborators at NHLI, and especially the Rose Tree Trust. And thank you for your attention.
Excuse me.
Use this, yeah. Hey, really interesting. Can you comment, how quickly can you get it to polymerize? Question one. Question two is, what about unreacted moieties? Is there toxicity from them?
Yeah. So it really depends on when you want to extrude the material, but the gel point, it's around 8 minutes after you cross-link them. So, and it's enough to have the material, enough time to have the material inside. So the There shouldn't be any unreacted moieties because we prepared the materials separately before. We dialyzed them thoroughly, so we have no unreacted product because they're dialyzed. So any moieties we have is attached to the polymer that then cross-links. If any of them doesn't cross-link, that's not a problem because they're not cytotoxic.
Last question there.
Yeah, my question was about, like you said, like the muscle-inspired chemistries. Was that changing here?
Oh, sorry.
Is that changing the, like in the microfluidic stream? So is that something to do with that, or you actually like loading with a different chemical to achieve that?
Yes, so it doesn't change the flow of the materials, but it fastens the cross-linking between the two because the muscle-inspired chemistry can adhere to tissue, but also to some gelatin polymer sequences. So the dopamine reacts both with the tissue but also with gelatin, so it just just fastens the cross-linking.
It's an extra like material that you're loading to the gel.
No, there's no extra material, sorry. The dopamine was attached to the alginate with the norbornine.
I see.
So the alginate was carrying both the norbornine to cross-link with the tetrazine of the gelatin and the dopamine to adhere to the tissue. Sorry, I didn't get
I
it.
see. Thanks.
Yeah, it was all on the same polymer.
I think we stop now for the questions, just the
Okay.
little time. Thank you, Nicola.
Thank you.
Thank you very much. And now we proceed with still another topic, bionic medicine. Laura? Yeah.
Hello everyone, my name is Laura, I'm a postdoctoral researcher in the Neuromechanics and Rehabilitation Technology Lab. So today I will talk about neural interfacing for individuals who have lost their upper limb. So before getting there, I just would like to remind ourselves of how we perform a voluntary movement. So the central nervous system coordinates the activity of several hundreds of thousands of sensory and motor nerve fibers. So in the motor nerve fibers, alpha motor neuron carry the information from the spinal cord to the muscle fibers. If we consider peripheral nerves, the axons are organized into fascicles. And if we consider, for example, the median, the ulnar, or the radial nerve that innervate multiple muscles in our upper limb, these are polyfascicular nerves, so they carry very rich information about several tasks of the upper limb. So after amputation, there is an interruption in the communication from the central nervous system to the muscle. However, the neural signals still go through the nerve. So targeted muscle reinnervation is a surgery that takes these polyfascicular nerve and reroutes them into available muscle targets that then will act as a bioscreen of the neural information. So once we have this reinnervated muscle, we can create a human-machine interface through electromyography. So ideally, to control a prosthesis, we would like to extract from the human-machine interface a number of signals that corresponds to the number of degrees of freedom that we have on the prosthetic. However, typically, these muscles are interfaced with a bipolar sensor, and for a muscle and a nerve, we can get out at most one signal. So it becomes very difficult to extract the multiple signals that corresponds to the different tasks of the limb. So to tackle this problem, another surgery that is called the regenerative peripheral nerve interfaces, RPNI, proposes to split the nerve into its fascicles that you can see here in yellow. And then each fascicle is attached to a little muscle graft. So we have 4 in this case, and they form 4 RPNIs. So each RPNI is then interfaced using a bipolar intramuscular electrode. So in the end, we get out 4 signals. So we have the potential to control 4 different tasks of the prosthetics. So this surgery has, however, a few limitations. So just to mention a few, the number of times we can split the nerve in 2 depends on the number of fascicles. And so there is a limitation in the number of signals that we can extract. Then we will know if the signal that we extract from the different RPNIs is actually of use for prosthesis control only after the surgery. So the nerve is split without knowing a priori the functional role of each fascicle. So within the lab, this problem of mixing the EMG signals and extract control signals for prosthesis control has been tackled in a way that is not surgical, so not by dividing the nerve, but purely mathematical. So high-density surface EMGs were used to extract motor units that are associated with the control of the limb, and high-density intramuscular electrodes were developed to obtain a more selective high-bandwidth signals, and these were tested in animal model of TMR. So we are proposing a novel biointerface that is based on this transfer of polyfascicular nerves and the use of microelectrodes array. So you can see in this concept image an amputee who had TMR, and he's performing different tasks with this phantom limb, for example, index extension. So at the same time, we record also the activity of the muscle. And you can see here in this zoom that the rerouted nerve have the axons that go and synapse with different fibers of the muscle, creating this distribution of neural signals that are then picked up by the different channel of the electrode. So what we hypothesize is that we can decompose these signals into a much higher dimensional space where each dimension is actually defined by the activity of individual motor units. And then we can cluster these motor units and associate them to specific function of the limb. So the electrodes that we are using are these ones. You can see 40 smaller channels distributed in 2 centimeters. And we record from 4 reinnervated muscle from 3 different participants. So all the recorded EMG signals are decomposed into the activity of individual motor units that you can see here color-coded. So for example, motor unit 1, that is in red, these segments correspond to all the instance of time where the motor unit is firing. So considering all the tasks and all the reinnervated muscle, we could decompose more than 100 motor units. And then once we have the units active for a specific task, we check if these units are task-specific or they are also active during different tasks. And what we see is that for each task, we have a cluster of motor units, some of which are task-specific in blue and some are shared. And only 3 tasks out of 23 had only shared motor units. So then we consider the space that is defined by the motor units. So we concatenate the neural activity, low-pass filtered, for different tasks of the phantom limb, so for the same reinnervated muscle. And we have 4 repetitions of the task, so this is why you see all these patterns, and we see that different motor units are active during different tasks of the limb. And then we hypothesize that this space can actually be explained by a number of latent components that is equal or smaller to the number of tasks that we measure. So we apply a simple method such as non-negative matrix factorization to estimate the neural manifold that is embedded in the space of the motor units. And when we look at this latent time-varying component, we can see that there are patterns of neural activation that correlates to the activity of the phantom limb. And if we look at the weighted contribution of the different motor units to a latent factor, we see that these units are actually clusters. So we could effectively decompose and extract different signals for these different tasks. And so just to conclude, we know that the polyfascicular nerve is carrying different neural signals to a muscle. And we have provided a proof of concept of a high information transfer biointerface by measuring only in a single side in the muscle. So I just would like to thank all the co-authors and collaborators at the Medical University of Vienna, the ReLimb and Royal Free Hospital. And this project was funded by Natural Bionics European project. Thank you.
Thank you, Laura. Are there questions? Please.
So maybe I missed this, but when you're saying, like, for example, you're doing a certain motion and then you're recording the activity, is that from the opposite hand and you then, like, assume it's the same on the other side?
No, so the nerve that was previously innervating multiple muscle in the limbs is taken and is rerouted into an available muscle that it can be the pectoralis, the latissimus dorsi, and then when the subject is performing a task of the phantom limb, that muscle is twitching. So we ask the participant to do that task, and then we record from that muscle. So
how are they able to do the task?
they send the command so they can imagine. So it's exactly, so imagine that you are able-bodied. If you are amputated, the only difference is that the neural signals at some point doesn't reach the muscle fibers.
I see. So they're imagining doing the activity and then you record
execute
which muscles.
actually the task. Yeah.
Yeah. Thanks.
Thank you. Thank you, Laura, again. The stereotype we continue. And next speaker is Anne. Is Anne here? Oh, yes, sorry. Please.
Hi, good afternoon. I'm Anne Go. I'm a staff scientist and facility manager at the Schultz Lab. And I'm going to tell you about this exciting project we're wrapping up in our lab. So the hippocampus is the part of the brain which is very important in learning and memory. It is critical for the encoding, the storage, and the retrieval of episodic memories. And it is also among the first brain areas to be affected by Alzheimer's disease. Now, in the brains of Alzheimer's disease patients, we see, among others, two things: intracellular neurofibrillary tau tangles and extracellular amyloid beta plaques. And we wanted to see how these plaques affect the neural circuits responsible for the encoding and the retrieval of memories. And so we sought to image the pyramidal cells in the hippocampus of wild-type mice and mouse models, a mouse model of early-onset Alzheimer's disease, 5XFAD. And we did that by doing 2-photon calcium imaging. So in our experiments, we image mice that are head fixed as they run around a circular track that's floating on air. And in the brains of these mice are JG-CAMP7S, which is a fluorescent calcium reporter, which reports the cellular activity. And we see an example of a 2-photon image we have here, the green in JG-CAMP7S, and the magenta is of methoxyXO4, which is a marker for amyloid beta plaques. So we can see where the cells are in relation to the plaques. And here on the right are the cells that our algorithm has identified for this field of view. And these are the calcium transients that we've obtained from the image. Now, one of the things that we saw is that the baseline firing, that is the activity rate of the cells when the animal is at rest, it's shifted to higher firing rates in old AD mice compared to old wild-type mice, as you can see in this CDF plots here on the left. We see a similar trend in the young groups, but our stats are not coming up as significant for those groups. Now, this has been reported in literature, but interestingly, we only see it in the resting phase. In fact, when the animals are running, we see the opposite. So you can see here in this CDF plot that the firing rate for the old AD is actually lower when the animals are running compared to the old wild type. Now, interestingly, the old AD mice actually run faster compared to the old wild-type mice. And here you can see that they spend more time in higher speeds. Now, if we plot the activity of cells against the running speed of the animal, we get speed tuning curves, such as this that's plotted here. And let's define the dynamic range as the maximum of this plot minus the minimum. What we find is that the dynamic range is decreased for old AD, so old 5XFAD mice. And I don't have time to detail the criteria, but we can use the dynamic range in the shuffling algorithm to identify speed-modulated cells. And what we find is that there are fewer speed-modulated cells in old AD animals compared to old wild type. There are actually 3 kinds of speed-modulated cells according to their speed tuning curves. There's the positive, negative, and preferred speed we observed. And when we look at the proportions at which they occur, we find that there are more negatively modulated speed cells in the old AD group. Now, because we know where the plaques are in relation to the cells that we measure, we can look at how the different properties that we measured change with distance from plaques. And what we find is, for example, here, the activity rate during rest is increased closer to the plaques. The activity rate during running is decreased closer to the plaques. And the dynamic range is reduced closer to plaques, which suggests that the aberrations in these properties seem to originate in the vicinity of plaques. One of the other things we look at are place cells. These are cells that fire at specific locations in the track when the animal is running. So for example, this is one particular place cell, and it only fires when the animal is in this particular part of the circular track. And what we find is that there are fewer place cells in the old AD mice. And here on the right, we see that the place field size, the width, or the fraction of the circular track where the firing occurs, it's actually reduced for the old AD. Now, if the tuning were degraded, we would expect it to be wider, actually. But we think this is because of the hypoactivity that occurs in these mice. Generally, we see a degradation in spatial tuning. So we see less spatial information encoded in the firing activity of the 5XFAD groups, both in young and old groups. The activity is less coherent in the old AD, and the activity is more variable from lap to lap in the younger AD group. It's a similar trend in the old group, but it's not coming up as significant. Also, when we compare the place fields in the first half of the experiment to the second half of the experiment, we find that they're less stable in the old 5XFAD mice. So NOV here is a novel environment. So a familiar environment is one in which the mice have been exposed to for a week, about 12 sessions. And the NOV is a completely novel environment they're encountering for the first time. And let me just concentrate on the panel here in C, and we look at the amount of laps it is required for the place cells to be tuned. In a familiar environment, there is a delay in place tuning in the old AD, as evidenced in this CDF plot here. But in a novel environment, both the young AD and the old AD have delayed place tuning. So there's delayed recall, and there's delayed encoding of new memories in this 5XFAD mice. So in conclusion, we have gained insight into the progression of this neural coding deficits in 5XFAD mice that affect the encoding of new memories and the recall of old. And oops, I seem to have lost my last slide there, but I'd like to thank collaborators in the lab and from University of Melbourne for all the help in this work. Thank you.
Questions? Maybe just a very brief one from me, very bit detailed.
Yes.
When you talk about coherent activity, what do you mean?
So coherence in space, yes, that's a good question. It's how close in space the locations of high firing rates are. Right. So the more coherent, the more closer in space, and the cleaner the place fields.
Okay,
yes.
But is there a measure of correlation as well, right?
Correlation, yes.
All right.
Yes, okay. In space.
Thank you. Other questions? Otherwise, we thank And again, thank you very much. And go quickly to John. Next speaker, yeah, please.
Yes, so hello, I'm John. I'm a PhD student in Dr. Andrei Kozlov's lab. And as a lab as a whole, we're interested in two areas of auditory neuroscience. We're interested right in the very beginning in the translation of sound energy into electrical impulses in the cochlea. And then we're interested in, right at the other end of hearing, the representation of complex sounds in the auditory cortex. And it's this second area which I'm going to talk a bit about today. So in particular, we often use the mouse's auditory system as a model organism. And we're interested in the representation of these complex vocalizations which the mice use to communicate with one another. And these are typically in the range of 20 to 110 kilohertz, so they're above our human range of hearing. And they're used for a range of behaviorally meaningful behaviors, such as in courtship, in fighting, and in interaction between the young and the parents. So as a stimulus class as a whole, this is a very rich and behaviorally meaningful set of stimuli with which we can probe the auditory system and its computations. Traditionally, USVs would be just recorded from a cage, but this is time-consuming, requires expensive equipment, and it requires manual processing and curation, all of which take time and money. So in this presentation, we present a neural network approach to generate synthetic USVs. And this promises to not only generate an unlimited set of USVs, but also, more importantly, it allows us much greater parametric control over how we generate these stimuli. And I'll give an example later as to why this is so important. So our network is based on an architecture called a GAN, or a generative adversarial network, which consists of two neural networks, a generator and a discriminator, which essentially work against each other to learn this low-dimensional representation of a set of stimuli, in our case, these USV waveforms. And we can also represent these waveforms as a spectrogram, and you'll see more of these today, where we've got time on the x-axis and frequency on the y-axis. Our model builds on this by being bidirectional, so it adds a third neural network, an encoder, which allows us not only to generate realistic waveforms from the latent space, but it also allows us to map both generated and natural USVs back into the low-dimensional latent space. And that allows us to answer some really important questions about natural USVs. For instance, whether they belong to discrete, meaningful clusters, or whether instead we should think of USVs as being a continuous spectrum. And if we try and cluster them, then we're sort of putting some human bias on their features, which we actually shouldn't do. And it's this latter explanation which we find in our data. So our model was then trained on around 30,000 natural USV syllables. And once we had the model generating realistic waveforms, we wanted to test whether they behaved the same in an actual mouse. So we recorded the activity of single units in the auditory cortex in response to our reconstructed USVs. And you can see here some single unit activity in the auditory cortex in response to two natural USVs in the first row and two reconstructed USVs in the second row. And you can see that the unit is firing in response to both the natural and the reconstructed USVs. We then used the data from all of the units which we recorded, from both the class of stimuli, to train receptive field models. And this allows us to predict the neural response to a section of unseen stimulus data. And here you can see, again, a spectrographic representation of some receptive field features using our reconstructed USV data. And what we find is that the receptive field features learnt using the reconstructed USV data are just as informative as those generated using the natural USV data. Not only are they just as informative as one another, but we can actually swap the training and the test data between the models. And the performance of the models is practically unchanged. And what this means is not only are the models just as informative as one another, but the receptive field features we've learned are essentially analogous. It makes no difference whether we use the reconstructed USVs or the natural USVs. And we also looked at some high-level response features, such as lifetime and population sparseness. And essentially, this asks the question of how selective either a single unit or a population of units is to a class of stimuli. And again, in brief, we find that these high-level response features are maintained with our reconstructed stimulus. So you might ask, if I've already recorded these USVs, why would I bother generating new ones? I could just play the ones I've already got, right? But as I mentioned earlier, it's to do with the control we have over how we generate these USVs. So looking again at our latent space, I can pick two positions in the latent space and interpolate between them. And this generates a smooth transition from one USV to another. And it is remarkable how smooth this transition is. And this is analogous to a network, for example, which can interpolate between a picture of a dog and a cat. And this is really important for studying things like decision-making in what the neurons respond to and what they don't. And here you can see an example of a single unit's response to one of these interpolated stimuli. And we can see just how precisely tuned the neuron is to selected features in the USV, with even small alterations in the USV structure causing a drastic change in the response. So the real power of our model is that given enough data, this is not just limited to mouse vocalizations. So we could theoretically train a model both on mouse vocalizations as well as human speech, and for that matter, whale vocalizations. And we could smoothly interpolate between vocalizations of these different species, and we can use this to study how the neural representation changes as we cross these categorical boundaries between behaviorally meaningful and behaviorally irrelevant stimuli. And with that, thank you for listening, and yeah.
John, the advisory board has to go, as we knew. There's a question there at the last row.
Thank you. The data that you collected in order to train the gun, right? The real mouse sounds. How many mouse was it recorded on? And also, when you made the mouse listen to the sounds, how many mice listen to those sounds? I mean, I just wonder how much risk there is to overtuning to specific mice listening or specific mice speaking.
Yeah, so we trained the model on 30,000 or roughly 30,000 of these syllables. They're publicly available, so they're not something which we've recorded. And then in the phase of the experiment, when we played it to the mice, we played it to 6 mice and recorded from a handful of cells, so a relatively large population of cells. So, yeah, again, I don't think there's any massive overfitting to the data. It's, yeah, generalizable.
Good afternoon, I'm Magdalene, and today I'll talk a bit about my PhD, which is interactive DNA nanobandages to promote wound healing. So wound healing is a very beautiful and complex process that uses multiple types of cells and growth factor to bring an opened, inflamed wound to a closed and remodeled wound. Some of the well-studied growth factors are PDGF, FGF2, VEGF, and HGF, which work together to bring about cell proliferation, vascularization, and migration, important processes for tissue repair. Unfortunately, for millions each year, wound healing is not as straightforward. Non-healing diabetic foot ulcers contribute to over 4,000 amputations a day. One of the reasons is that For these wounds, there are often a deregulation of growth factors for sufficient wound healing. Hence, a strategy is to supplement these wounds with additional growth factors, but current delivery strategy through a spray or a gel is highly inefficient with nothing to hold the proteins in place. As it travels through the body, it might cause unwanted tissue growth, increasing the risk of cancer. Hence, there is a huge need for targeted delivery of growth factors. To tackle this, we turn to nature for inspiration, tracing as far back to sea urchins as an evolutionarily conserved delivery of TGF-beta-1. So TGF is deposited in the wound in a cage complex, and it is released and activated in response to cellular traction force. Mimicking this, we developed a synthetic version called traps. Traps are made of single strands of DNA that fold and bind this protein of choice. And when cells at the wound actively pull and grab on the trap's handle, it causes trap to unravel like a shoelace, releasing this protein to accelerate healing. This precise on-demand delivery is like unwrapping a gift exactly when you need it. To validate this system, I seeded endothelial cells in a spherical shape and placed it in a collagen gel. As a control group, I have scrambled traps, which are basically traps with a non-functional handle, which means that cells cannot pull on it to release the growth factor. In this case, the therapeutic is VEGF, so you would expect to see a lot of endothelial sprouting to initiate blood vessel formation. And it's exactly what you see. And if you look at the traps group, it is promoting thicker sprouts than the scrambled traps group. And if you look across timelines, especially at 24 and 48 hours, you do see that traps promote longer, thicker, and more sprouts. This is an indication of early vessel initiation and progression, suggesting that we're successfully able to deliver this VEGF as expected. The next question is then, can we maintain this functional integrity in actively repairing tissue in vivo? So we did an in vivo study where we took made a 6 millimeter gap in a rat's femur, and in that gap placed either an empty collagen sponge or a sponge functionalized with traps. So we harvested the bone and made cross sections of it to quantify the blood vessels after 3 weeks. And when you look at the traps group, it promoted many bigger vessels than all control group, which is exactly what we wanted. Notably, when you look at the scrambled traps group, it resulted in many small vessels, further suggesting that it was unable to release these growth factors, and so there was unlikely any passive release due to degradation. So yes, we can do it in vivo. This targeted delivery enables up to 1,000 less proteins than existing clinical strategies, making it a lot safer and more cost-effective. The next question is, can we completely eliminate the reliance on manufactured, expensive proteins that competitors rely on? Can we instead harvest these growth factors directly from a natural source, such as blood, at the point of care? In this way, we can harvest multiple types of growth factors for holistic tissue healing, potentially through a medical device pathway. And so, that's what I tried. I harvested FGF2 from blood lysate and showed that we were successfully able to do this to promote fibroblast proliferation. Likewise, we were able to harvest PDGF to promote proliferation and HGF to promote endothelial cell migration. So we have a system that can harvest or selectively harvest multiple types of protein therapeutics that can target multiple types of cells. And so I wanted to validate this in a translationally relevant ex vivo human skin model. So I took skin from tummy tuck surgeries, made a wound, and placed a sponge in it. As you can see, after 8 days, the control, the sponge in yellow, you don't see much happening. Likewise for the scrambled traps group. But when you look at the traps group, you do see increased integration of the epidermis, which is the cells are in magenta, and the cyan color is DAPI for the cell nuclei. So you do see increased epidermis migration towards the scaffold. And if you look at the cross sections, you do see increased dermal integration, especially in the group with 3 different types of traps. And when you slice the wound in the cross section, the group with the combined traps were able to, or had a higher probability of remaining intact as compared to all other groups, suggesting that there is increased cell infiltration that supported a more robust interface. So using the dermal integration as a matrix for the speed of wound healing, we showed that traps were able to promote faster healing by up to 2.5 times compared to the control group. I further validated this system in an in vivo mice skin model in collaboration with the University of Arkansas. And so when you look at the combined traps group, it did promoted smaller wounds as well as greater healed tissue. And so in conclusion, we're able to show that this targeted delivery can increase efficacy while significantly decreasing the therapeutic dosage needed, making it a lot safer and more cost-effective. So I'd just like to acknowledge my lab, the Elmquist lab, and everyone highlighted contributed to the animal studies. Thank you.
Thank you very much, Magdalene. Very clear questions. Maybe one question is, sorry.
Could you expand on your methodology for the mice skin test, the one you did in collaboration with Arkansas, please?
What do you mean about methodology? We did a full thickness skin wound.
Right, okay. But how many tests did you make? Did you do, sorry, for the mice?
So we did 3 treatment groups, N equals 4 for day 4, and N equals 6 for day 10.
Okay.
And half female and male.
All right. One more question, please.
Like, you mentioned the fact that the forces made by the cell is what stimulates the molecule to get removed from the trap. Like, how do you know what kind of force? Is it dependent on like different cell types? Like, I don't know, different types of skin areas will produce different forces, and then to be able to, I guess, design the right kind of trap molecule for the right type of force.
So you could, but in this case, we're using the very low force, so forces when cells migrate and directly pull on the handle. So you could also tune it to be more cell-specific in terms of force, but you could also change the handle to target specific type of cells.
But right now it's just general, so any kind of movement would cause it to be released?
Any kind of engagement with the handle.
Okay.
This one we use RGD, but in the previous paper we use one that is specific to different cells.
Thank you.
Thank you. Thank Magdalene again. And next is Leo. Yeah, it is talking about eczema. Please.
All right. Hi, everyone. My name is Leo. I'm from the Computational Systems Medicine Group, supervised by Professor Reiko Tanaka. And today I'll be presenting a high-level overview of the exciting research going on in our group. So this talk is titled Automated Evaluation of Eczema Severity Scores for Diverse Skin Tones. So I'll start by talking about eczema. Eczema, as many of you might know, is one of the most common chronic inflammatory skin diseases worldwide. It results in a high socioeconomic and quality of life burden. It's typically manifests as spatially distributed lesions over the entire body and can result in various symptoms such as redness and dryness, each of varying severity. So in our group, we're particularly interested in assessing overall eczema severity, since this is important for deciding which treatments are required and also if a specialist referral is needed. So in particular, we are interested in remote eczema severity assessment. Now, the motivation for this is twofold. So firstly, since the COVID-19 pandemic, many more consultations for skin and eczema are taking place online by sending in digital camera images to doctors and clinicians. And secondly, studies have actually shown that remote eczema severity assessment correlates strongly with in-person severity assessment, so demonstrating its potential for facilitating remote care. So this is a typical example of what severity assessment via traditional teledermatology might look like. So we begin with a patient who submits a photo of their skin via a mobile or web application. This will then go to a doctor who will review this information and then use this to make an informed clinical decision about the patient's next stage of care. There are a few problems with remote eczema severity assessment. So one major problem is that of inter-rater variability, which basically means that different clinicians might assign different severity scores to the same image given the subjective nature of the task. And this is especially true in patients and images of darker skin. So we have proposed to use AI to try and address this issue. So as you can see here, we add a single step to our previous workflow where we have an AI-based model predicting the severity, and alongside the image, then the doctor will take all of this information to help them make a more informed clinical decision. There are, however, some additional challenges that come with using AI. For example, current models are not quite robust enough to be safely deployed in clinical settings. So to summarize the overall goal of our project, we want to develop a robust and reliable AI-powered tool that can assess eczema severity from digital camera images and deployable for clinical usage for patients of any skin type. So what would such a model look like? Well, I present to you a model called ExmaNet, which is a convolutional neural network-based pipeline for eczema severity assessment previously published by our group. So I'll walk you through how it works. We start off with an input eczema image, such as this. And firstly, we pass this image through a region of interest detection model, which extracts crops from the image that contain eczema regions. We then pass each of these crops through a convolutional neural network-based severity prediction model to output severity prediction scores. So this makes probabilistic predictions of 7 common disease signs which are typically seen in eczema. And then we integrate these scores from the 7 different signs to result in an overall regional severity score for a given image. And then later on, we develop an enhanced version of ExmaNet called ExmaNet 2. So it works as follows. So we start off again with the sample image, and this time we replace the region of interest detection model with a pixel-level segmentation model based on UNet. And this can either segment the entire skin region or just the eczema or atopic dermatitis region, as you can see here. Then we have some more post-processing steps. So we start off with a border following algorithm, which extracts rectangular crops from the skin or eczema regions, followed by a square cropping algorithm that extracts square crops without distorting the image. Finally, this goes to a severity prediction model similar to ExmaNet 1. So there's a couple of ways that we are trying to further improve the accuracy of our model now. So one of these is through an image quality assessment model, which we would place prior to the severity prediction model in the overall pipeline. And this is important because AI models are typically trained on high quality images, and therefore they assume high similarity between their high quality inputs and any new data that they face. However, if we imagine that this kind of model would be deployed in a real clinical setting, the quality of these kind of images, which may be acquired by patients or clinicians, we can't always guarantee that these images will be high quality. So if a low quality image is inputted into the model, this might result in inaccurate downstream predictions. So we propose to use an image quality assessment model to, number one, screen out low quality images, and number two, to provide prompt user feedback on how to improve the photographic quality of their input image. So what would this look like in practice? Again, we start off with a patient submitting a photo. We would pass this photo through our image quality assessment model to output a score for image quality. We then check if it meets a predefined threshold. If it doesn't meet this threshold, then we would also output a classification. So what is the kind of distortion in this image causing it to be low quality? For example, motion blur. And then based on that distortion, what can be done by the user to improve the quality. So in this case, it would be a simple case of just keeping the camera still. So we feed back this image, we send this feedback to the patient, and then they can take this on board to resubmit the photo. And this process repeats until finally the outputted score meets our given quality threshold. It can then be passed to the downstream AI prediction model, in this case, which is for severity prediction. So overall, our goal is to try and integrate all of these modules into an end-to-end pipeline, which can be deployed for clinical usage and for mobile application. So it would look like this. We start off with our input eczema image and input quality checks. Then we would assess the eczema severity, as well as potentially detecting whether any viral infection is present in the image. We would do some output quality checks, and finally we would output the final score to the patient. So just to sum up on a couple of other ongoing research areas in our group. So, okay, well, these are the ongoing research areas. Okay, so one of the things that we're doing is mechanistic modeling to try and uncover the disease mechanisms of eczema. We're also trying to incorporate uncertainty quantification to improve the trustworthiness of our models. We're trying to generate synthetic images to address the problem of data scarcity, and we're trying to use our generated severity scores to predict future eczema flare-ups via time series models. So thank you very much to my colleagues and my funders, and thank you.
Leo, question here.
Thank you so much for your presentation. Very, very interesting. I had a question on the way that the assessments of the individual patches are basically combined. So do you run your assessment on the eczema net sequentially, and then you produce a combined score based on all the scores that you get from the individual image passes? Or is there any way that, for example, if different combinations of things that you see in different passes would mean different things in combination afterwards, to also take those into account?
Yeah, thanks. Good question. So it's kind of, we assess the severity of all the patches together, and we kind of have, we measure the same things for each of the patches. So as I said, we have 7 different severity signs. Each one has a score. It would be an ordinal score between 0 and 3. And then based on each of those scores for each patch, we do some kind of a convoluted sum to find the overall score. So it's the same process that's done for each image.
But there's nothing implemented that, for example, well, if you have a green spot here and a yellow spot there, this actually means that the outcome should be something different. So there's no recombination of that information happening afterwards.
You mean specifically for a feature like color, or?
For example, it was a bit bad description, but that's basically what I mean.
No, so we don't have that. We are only looking at these specific clinical signs. But yeah, thank you.
There are more questions, but in the interest of time, we'll have time to network after. We thank Leo again.
Thank you.
And, yeah, you will have time to talk to Leo in the break after the session. Larry is the next speaker on millifluidic approaches, please.
Hi, folks. So it looks like I'm the only one who didn't get the message about the standardized templates, so I'm very sorry about the aesthetic choices I'm about to subject you all to. So, yeah, my name is Larry O'Connell. I'm a postdoc in the group of Daryl Overby, and today I'm going to be talking about millifluidic approaches to bulk tissue perfusion. Okay, so as an overview, first I'll be talking about why we might want to achieve nutrient perfusion through tissue ex vivo, and then a few methods we've developed to pursue this. Okay. So when you resect tissue, either through a surgery or from a sacrificed animal, oh, sorry, give me a minute. Huh? Oh, 1 second. Sorry, it's a problem with the formatting. Okay. Okay. I think we're good. Okay. Great, thanks. Okay, so when you resect tissue, either through surgery or from a sacrificed animal, you sever the native vascular network, and you remove the native vascular system that delivers oxygen and nutrients, and you remove the system that removes metabolic waste and CO2. And so if you don't have a strategy to replace that transport system, the tissue dies, okay? So any system that aims to maintain viability ex vivo will need to replace this transport network to keep cell metabolism functioning. And so the current gold standard in tissue viability maintenance is culture of these thin tissue slices. So essentially you rely on just diffusion. You replace the vascular transport network by cutting such a thin tissue slice, about 200 micrometers thick, that all of the cells find themselves within about, at most, 100 micrometers of the nutrient medium. And so then they can thereby benefit from nutrient delivery just by virtue of diffusion. Okay? As the problem is that this fails to capture the vascular transport dynamics that we see in vivo, and any therapeutic agent that is introduced to the medium is going to have total access to the entire tissue. Okay? So it's bypassing the vasculature. And so if we hope to create a model system that recreates the vascular transport dynamics, we need to perfuse the tissue via the native vasculature. And the payoff, if we can achieve this, is that by retaining the native tissue microenvironment, this will enable better drug testing, personalized medicine, for example, in chemotherapy, which is what my postdoc focuses on. Reduction in the number of animals used if multiple tissue biopsies can be sourced from each animal, will also allow further fundamental research into vascular transport phenomena. So a very quick note about the difference between perfusion and perifusion. Perfusion drives fluid through the blood vessels of tissue, while in perifusion the medium flows around the explant. And then we're just back to diffusion regime. So the first model we developed was based on a hydraulic constriction, which is a cylindrical tissue biopsy, and it's introduced into a millifluidic constriction, which causes a self-sealing system whereby the cross pressure coming in from the left does double duty, right? So first it creates compressive forces on the periphery of the biopsy to block off perifusion routes around the tissue. And second, the cross pressure drives flow through the capillary bed of the tissue biopsy. So if we spike the perfusion medium with fluorescent tracer beads, we can visualize the routes of fluid flow through and around the explant. What we've found is that although there are some beads delivered through the bulk of the tissue, the majority is flowing around the edges, okay? So this is perifusion, which you want to avoid. So there isn't effective delivery of beads throughout the biopsy. And so even though we get a little boost in terms of viability markers, we're clearly not achieving good intravascular flow. So the next model we developed relies on cyclic compression of a tissue biopsy. The idea here is pretty simple. You carefully apply a compressive force to the entire tissue and you hope to actuate the native vasculature. And so during the compression stroke, the vasculature, as the most compliant part of the tissue, essentially voids within the tissue. They compress and eject their contents into the surrounding medium. And then during the decompression stroke, the vasculature redilates, drawing fluid from the surrounding medium and into the center of the explant. And that way we can achieve advective mass exchange with the explant core. So this is a confocal image through the center of a tissue biopsy that was subject to cyclic compression. So again, the perfusion medium has been spiked with fluorescent tracer beads to visualize the advective nutrient delivery to the explant core, see if we can beat diffusion. And what we can see is that there are some perfused branching structures reminiscent of vasculature, but we still have these very large regions of unperfused tissue. So to improve on these compression models, we're taking a new approach whereby the perifusion routes are cut off using a proprietary method, which I can't reveal yet, all along the periphery of a comparatively large tissue biopsy. So the tissue biopsy is about 8 millimeters wide, 3 millimeters thick, so it's kind of a puck shape. And since the perifusion routes are cut off around the tissue, we can simply add a cross pressure to drive perfusion across the entire volume of quite a large tissue biopsy. So this is a confocal image across one of these wide liver tissue biopsies that we perfused in one of our latest devices. It's about 5 millimeters across, and again we've spiked the medium with these 200 nanometer tracer particles. And the peripheral regions we disregard as they're not really expected to be perfused in a way that leaves the liver tissue in a state that's easy to interpret. But instead we focus in the central region, in the blue circle, where there's been a simple cross pressure into the plane of the image through the tissue and out the other side. And what we see is there are these tubular structures that traverse the entire 2 to 3 millimeter thickness of the liver biopsy. And the tracer particles reveal the perfusion routes generated by our device across the entirety of the tissue. And they're spaced every few hundred micrometers or so. And so we expect that these perfusion routes through the tissue will allow gas exchange, nutrient exchange, waste removal, et cetera, from the entire bulk of the tissue. And so the next step was to confirm these perfusion routes are vascular. So we spiked the medium not just with tracer beads, but also with fluorescent isolectin 649, which helps visualize the endothelial lining of the vasculature. And now we see co-localization of the tracer beads with these tubular structures revealed by the fluorescently tagged isolectin. And this is a planar section across the face of the biopsy, about 1 millimeter into the tissue. And in comparison, here's a transverse section from the same experiment, visualizing the perfusion of medium through the entire thickness of a macroscopic tissue biopsy. So the flow is coming from right to left. And so you can see that this is an 8 millimeter by 3 millimeter tissue biopsy where we've restored true perfusion of nutrient medium throughout the bulk of the entire tissue. Next steps will be to confirm the extension of viability using histology for various metabolic markers. So in summary, to accurately recreate the tissue microenvironment, the best approach is to take a mature tissue and restore vascular nutrient delivery. We're pursuing this with a variety of approaches. Next steps will be studying metastasizing cancer cells, both at the stages of intravasation, so that's leaving a tumor biopsy into the vasculature, and at the stage of extravasation, where these circulating metastasizing cells are colonizing new tissues by leaving the vasculature. So I'd like to thank the PI of the group, Professor Overby, and also postdoc King Asuba, PhD candidates Severio Showerlambos, is that right? And Eva Zoringa. Okay, thank you.
Thank you, Larry. Questions? Larry, all clear? So maybe we take advantage of this to record a few minutes, and we thank Larry again. Thank you. Fabian, next, animal behavior, please.
Thank you. All right. Hello, everyone. Hope you guys are still awake after these fantastic presentations and have some capacity left for the stuff that I want to show you today. Hi, my name is Fabi. I'm a postdoctoral researcher at the Evolutionary Biomechanics Lab with David Labonte. Today I want to talk about from 3D models to 3D insights, specifically how advancing animal behavior research is possible with open source technology. First things first, biology is messy. Not necessarily the animals that we work with. These ants actually clear up very, very nicely after themselves. But what I mean more is, in biology, we're moving from a state of doing everything in a more or less qualitative way, where a couple of decades ago, an ethogram and describing some behavior just purely visual was enough, to a state where now we want cold, hard data. We want ways to look at video footage and be able to tell exactly how animals move, how animals cooperate, and how they're able to do very, very difficult, well, energetically difficult tasks together collaboratively. Now, it seems like in all things data science these days, we just try to throw machine learning at these problems and hope that this kind of sorts out itself. But if you want to get things like accurate 3D motion, or also how these animals interact, it's not necessarily trivial if you want to do these things in naturalistic environments. Because whenever we train these models, they might work well in one kind of static setting, but the second we change anything about the appearance of this video footage, the quality of the inference that we get deteriorates drastically. And that basically means that things like this pose estimation that you see of the stick insect over there, or these ants that we kind of track around, all of this gets very, very bad the minute the lighting changes, the camera angle changes, magnification changes, the species changes, or the application changes. So when we think about the way that we need to train these networks, which mostly happens in a kind of transfer learning approach, meaning we start out with a network that has been trained on something else, and then we refine it on something like the key point estimation or the bounding box estimation, if we now want to switch tasks and also figure out, well, what kind of poses do these ants actually take on, well, then we just need to start labeling again. And that just becomes very, very labor intensive. One potential way out of that is the use of synthetic data. And we've heard that quite a few times today already, and we're not taking the approach of generating them through the means of a neural network, but instead we do this procedurally with a gaming engine. But what do we need for that? The first thing we need is a nice 3D model of our study organism. In this case, we have an Atta vollenweideri over here. It's just a type of leafcutter ant that we work with. We then want to be able to simulate as much diversity in our training data as possible that basically simulates the variability that we would find out in the real world, so we can take these tools and not only have them work in laboratory conditions, but ideally also when we are in the field. And then once we have these training samples, we can then start training our neural networks again to hopefully get performance that looks something like this. So let us start with the 3D models. Where do we get them from? Well, scanning very, very small things is very, very hard because you ideally need as many different viewing points of these animals as you can possibly get in a way that doesn't damage the animal and makes it possible to run something like photogrammetry, so taking these viewpoints together and then reconstruct a 3D model out of that. And because there just wasn't a device available that made all of this possible easily, we started to make something like this ourselves. This one over here is Scanned. It's our open source macro 3D scanner. What this one basically does is you see these two arms moving around the subject in there. There's this big lighting chamber, and you have the animal inside that rotates back and forth. But you need one more degree of freedom here that also does focus stacking, because as you can imagine, if you've ever looked through a microscope before, things are very, very shallow in the depth of field. So we need to combine multiple images together in order to get a single sharp image out of that. This is kind of the process that we outline over here. So you get all of these different viewing angles. You then take these images and combine them together to get a nice stacked sharp image. And then you also need to remove the background, because otherwise photogrammetry software that you would run after this will get confused about something that moves in front of a background that is static. Because photogrammetry, or structure from motion more specifically, essentially assumes that you have a camera that moves around in a scene rather than an object that rotates in front of it. But with all these challenges in mind, and actually overcoming them luckily now as well, we end up with 3D models that look like this. So we can get really nice and articulate 3D models of the insects that we can put inside, and it's possible to scan anything between 5 millimeters and 50 millimeters in length. That opens up quite a bit of flexibility in the types of things that we can put in there. And when we actually released the paper on this, because we were all about accessibility and making sure that other people can replicate these successes with other things that they might want to scan, they did. And to this day, we have about 50 people and institutions that have come back to us that have built their own scans. And this is my favorite slide of all time, because it shows all the different people using their scanners for different applications. But just from the activity that we see online, it's very likely that there's way more of them out there, and on at least 5 continents that we can see just from the activity. And because so many people have come back to us and basically said, can you just make one of these devices for us, because we don't have the time and the 3D printers laying around to make them, we're now turning this into a little spin-out company as well. Let's see how things go there. Let's talk about 3D insights. We now have our high-resolution 3D models, and now we want to do something with it, which is generate synthetic data. In our lab, we developed Replicant, which is just another fun pun that we came up with. That is a software that we've written in Unreal Engine 5. Now, the reason why we've chosen Unreal Engine 5 is because you might have seen it in video games before. It's fantastic. It runs on pretty much any consumer-grade hardware, so it makes it possible to generate something photorealistic with very, very little cost, like all the other endeavors that we're also trying to capture here. This basically works by building this world procedurally, so you wiggle around that ground plane, you populate the world with all sorts of 3D assets, all of this is integrated in the software, you place some materials on top, then you populate this world with your animals that you want to learn from, and finally you render out an image. Now luckily all of this is entirely automated, so it's only a couple of mouse clicks that you actually need to perform in order to use this pipeline. And what's even better is that unlike the example before, where we needed to hand-click lots of stuff and do this once and repeat this every single time anything changes about our recording conditions, all the different post data and the location data and the 3D data of the mesh is integrated into every single frame that we render out. So a single 3D model can form the basis of training for lots of different applications downstream. All right, let's see if this plays. So here we have two of these applications. On the left side, you can see a stick insect. In the beginning, I hope the dots are big enough. This is key point estimation with just the model being trained on synthetic data alone. And now if we show that we just add 5 hand-annotated samples, we get pretty rock-solid pose estimation in there as well. And on the example in the right, this is something that a friend of mine kindly recorded for me when he was out in the Amazon forest, actually. Let's see if we can play this again. There we go. In this case here, the network has never seen a real ant before. This was entirely trained on synthetic data alone, and it also makes it possible to run this kind of inference when it visually changes quite a bit, so you see the changes in lighting condition. Finally, to say, what do we do with all of this now? Well, one big application is robotics. We want to learn how these animals walk and behave in 3D space to better inform how do we need to design 6-legged robots and how do we need to also model their locomotion efficiently so our robots can also, just like the animals, start climbing up our walls a bit better. And on the other side, we use this for large-scale behavioral studies where we have tens of thousands of animals that freely forage to track their behavior around in terms of what do they do in terms of their pose, what kind of actions do they perform, and do they actually operate as efficiently as literature would like you to believe. Quick conclusion, synthetic data can power robust machine learning. High-resolution 3D models form the basis for this, but there's many other applications. And open sourcing these tools has driven accessibility to a point where now people come back to us and share their innovations back to the original makers of the scanner. Thank you very much for your attention. Thank you to the, well, now expensive evolutionary biomechanics family, and thanks.
Thank you, Fabian. Very exciting research. Questions?
How long did it take you to learn how to use Unreal Engine?
It took me a while, but luckily I also had some help because when I originally pitched this idea, a friend of mine from high school had just learned to become a game designer. So he was already very proficient in Unreal Engine 5, and because he had some free time besides a couple of other projects, we started the work on that together so he could teach me in the beginning, because otherwise it is a rather steep learning curve when you come from more linear programming.
How expensive is this scan?
So the scanner is built in a way that you can also strap your own camera on top, so it can be relatively cheap. If you just go for the bare bones kit, it's about 1,000 to 1,500 pounds. If you want the fully integrated version that comes with its own camera and software and all the processing, that's about 4,000 pounds at the moment.
Just a brief comment. I was thinking the scanner used for educational purposes. So have you got in touch with museums?
Yes, yeah. We've gotten in touch with loads of museums at this point that want to use this kind of technology to scan their vast collections. It's quite beautiful. We've been flown out to Madrid and to Vienna when they now use these scanners for mostly educational purposes, for outreach, and also for conserving animals that are holotypes, so the ones that you really do not want to break, but still want to share information about in terms of morphometrics, for example. That's exactly where this comes into play now.
Well, very exciting. We thank Fabian again. Thank you very much.
Thank you.
And, last, Freya, please come to the stage.
Thank you. Today I'm going to talk about the Advanced Longitudinal Cohort Study. So ADVANCE is a 20-year longitudinal cohort study, and it involves 1,145 men who served in the Afghanistan war. Half of these men were exposed to a severe combat trauma requiring aeromedical evacuation to the UK, and the other half are matched controls who also served in Afghanistan. Of those who are exposed to injury, we have a large group of over 160 people with limb loss, which I'm going to talk about a little bit later in my talk. So as I said, it's a longitudinal study. We'll be collecting data from 6 time points over 20 years. Most of the injuries occurred between 2003 and 2014. We started collecting data at baseline, which is an average of 8 years after the initial injury or their match to deployment. We have baseline data, we have first follow-up data 3 years later, and we're 50% of the way through collecting follow-up 2 data, and we'll have 3 more time points to go, taking us to 20 years. This study is conducted by a huge team. It's a collaboration between King's College, Imperial, and the Academic Military Rehabilitation Centre, and in Stamford Hall, there is a large team of research nurses and additional personnel collecting data from our participants on all sorts of things: mental health, respiratory, traumatic brain injury, social support, proteomics. The data that I look at is our musculoskeletal data, which includes knee and hip radiographs, full body DEXA scans, which is going to look at bone mineral density, and lots of questionnaires. I also collaborate with other members of our team to incorporate gait biomechanics and serum biomarkers. So first I'll talk about knee osteoarthritis. So knee osteoarthritis can be graded radiographically using a Kellgren-Lawrence scale from 0 to 4. So 0 is a nice healthy knee, and 4 is end-stage disease, potentially requiring joint replacement. We define osteoarthritis typically from grade 2 and above. So at baseline, all of our injured participants had increased odds of having KL2 or above. The knee-injured groups, if they had a specific knee injury, a dislocation, a cruciate ligament injury, they had an 8 times increased odds of having KL2 or above, which fits with what we know from sports literature. Those with lower limb loss had 2.7 times increased odds, and even those who did not sustain a specific knee injury and did not have any lower limb loss but were still injured in another way, they still had more than 1.5 times odds for osteoarthritis 8 years after their injury. And then we followed them 3 years later, but what we found is that there was no progression, or there was no different amount of progression. They all progressed a little bit, but everyone progressed at the same rate as the uninjured group. And then for incidence, which is new osteoarthritis within that time, the knee injured and the non-amputees, they were the same as the uninjured, but our lower limb loss group had 2.5 times increased odds of having new osteoarthritis in that time, which led us to develop this model that assumes on the black line that we expect there to be a decline in knee health from midlife in the general population. So what we think we've seen that we measured at baseline is in the red line, so a dramatic decline in knee health earlier than we would have expected otherwise. But then in purple, it continues to decline at the same rate as an uninjured population. And then through what we show with those with limb loss is we hypothesize that because they have altered gait biomechanics that continues to affect their joints throughout their life, that they decline, they continue to decline at a steeper rate. And at the moment, we're investigating those with limb loss as a subset. And in the literature, it suggests that unilateral amputees have more osteoarthritis on their intact side knee. And work from my lab has shown this with biomechanical studies, that there's increased joint contact forces on the intact side limb. What we didn't expect to find in the advanced cohort is the Kellgren-Lawrence score is the top 2 lines. So this is just looking at 41 unilateral transtibial amputees. So they all have 2 knees, an intact side knee and an amputation side knee. But at the moment, so 11 years after limb loss, the amputation side knee is showing higher Kellgren-Lawrence scores. And we are collaborating with gait biomechanics specialists in our lab because for 11 of those 41, we have the motion capture associated with them as well. So we can combine that data to actually understand what's happening in these individuals. And that's never been done before in the literature. So we're very excited to be able to work together on that. Another thing we've looked at is upper limb disability using a questionnaire. So we showed, as we would expect, our participants with upper limb loss had worse upper limb disability, more than 8 times. More than 8 times the odds for it. But what was surprising is that there was no difference between those with a major upper limb amputation, so higher than the wrist, compared to those with a partial upper limb amputation, so involving the hand, thumbs and fingers. And this was seemed to be particularly impacted by the thumb. But I will say that all but 2 of the major upper limb amputees used prosthetics. So this is when wearing a prosthetic device. We've also looked at the effect of low back pain and low back pain related disability in those with lower limb loss, because we know that there's lots of biopsychosocial factors that affect people in the general population, but lots of those biopsychosocial factors affect people with limb loss, so we wanted to know how those things converged. And we found that when they had lower limb loss with with more low back pain related disability, often heavier, currently taking opioids, they actually were more likely to have experienced a period of low back pain before their limb loss. So it was sometimes might have been a recurrence. They had worse depression and worse phantom limb and stump pain scores, potentially due to altered nervous processing. And the advanced study is just one bit of work that our lab is involved in, and it's really key to demonstrating how lots of these interdisciplinary methodologies can all come together to create impacts in a real population, and sort of advice for rehabilitation outcomes, so using lots of computational studies, gait biomechanics, rat models. We do lots of work with children and the advanced study as well. So thanks to my lab and everyone on the advanced team and the funders.
Thank you, Freya.
It's not a scientific question, sorry.
Please.
With something as so long term, how much support does carrying out a study like this take in terms of like?
Yeah, it's an absolutely huge study, and it's definitely not something. It's definitely benefits from being a collaboration with the Military Rehabilitation Centre, with King's and with Imperial. There's a huge team of people who collect the data, administration, processing the data. I get sent an absolutely wonderful spreadsheet with all the data on. That's lovely to work with, but it doesn't just sort of get there overnight. So yeah, and then there's all the different specialities and different types of data that we collect, that there's another person like me doing the same thing. So it's vast.
Good. Thank Freya again. Thank you very much.
Thank you.
So we're finished. I would like to thank all the speakers. I was very impressed actually by the quality of the presentations and the variety of topics. I've learned a lot. I'm sure you have as well. So it's something definitely to repeat. That's a suggestion for the department. Thanks to Kemi for organizing the logistics, Calypso for organizing the scientific program. All of you to attend, please stay. There is a little party with some food and snacks so that we can all discuss and talk again to the speakers. Thank you very much, everybody.